Good evening, all. Today we are going to learn about a new topic which is very interesting, which is the Lytic cycle and Lysogenic cycles of phages. And in previously, I will remember one word, that is the bacteriophage, which means the virus that is infected to a bacteria is known as the bacteriophages. That means inside the bacteria, the virus can be seen. So let us study in detail. The virus that infect bacteria are called the bacteriophages, and there are two different cycles that bacteriophage may use to infect their bacterial host. Okay, and the two cycles are the lytic cycle and lysogenic cycle. In the lytic cycle, it is the lys, which is the breakage is happening, and the first itself there is shown a diagram which is the lytic cycles, and we can see the flow chart or the cycle way of the lytic cycle. The first is the attachment and inject viral genome into the cell. The viral genome is inside the cell is happening, and the second stage is that the degradation of host genome. Okay, then the third one is the replication of viral genome and synthesis of the viral proteins. And inside the cell itself, the viral genome is replicated, and the new one is the synthesizing or the viral proteins. Then is the which is the assembling and the release of what? The genomes. Likewise, the death of the host cell is happening, and the new viruses are producing. Okay, and that is the bacteriophage. From the bacteriophage itself, it is happening the cycle, that is the lytic cycle. The lytic cycle is commonly referred as the reproductive cycle of the bacteriophage. Okay, the name itself, it is a reproductive, means reproducing new ones. Okay, and a virus undergoes lytic and lysogenic cycle to reproduce. The virus is undergoing for lytic cycles and lysogenic cycle for the reproduction. And it is relatively more common wherein a virus infects a host cell, its metabolism to multiply and destroy the cell completely. So we in the previous picture we had see that the host cell is degenerated, or it is completely dead, and the new bacteriophages are new bacteriophages that infected virus containing bacteria is producing new ones or reproducing new ones. In lytic cycle there are 5 steps. One is attachment, second penetration, third synthesis, fourth assembly, and fifth release. The first one is the virus requires a host cell for multiplication. So the virus cannot multiply itself. So it's needed a host cell. So for that, the virus requires a host cell for multiplication. In a first step, the virus attaches itself into the host cell. First, it is not seen in individually, it is attached to someone, the host cell, and the virus may attach itself on the receptor of the cell or simply any other parts on its surface. In some cases, a small part of the virus attaches to the host instead of the virus itself. Okay. Then the second one is the penetration. Once the virus gains access to the cell body by breaking through the cell's plasma membrane. Once the virus is into the host cell, it starts its process by breaking its cell plasma membrane. Okay, then in some cases the cell wall tail fibers of the virus and enzymes released by its help in breaking through the same. The virus then releases its genetic material into the host cell. So finally the virus has released its genetic material into the host cell. For what? For the infection, for infecting the host cell, or for the degradation of the host cell. The cell is now said to be infected. And third one is the virus multiplication, cell destruction. Now that the genetic material as well as the viral parts are replicated, the host metabolism assembles new viruses from the scratch. From that, the genetic materials, the viral parts are replicated. When the virus is inside, from when the virus is inside the host cell, it starts replicating through what the genetic material starts processing and replication ongoing. The capsid is assembled, followed by the genetic material in the head, and finally the tail fiber is attached to the end proportion. Eventually, the host cell is filled with viruses. The host cell is completed when the virus is replicated again and reproduced again, and it is whole. In the whole, the cell of the host is filled with viruses. Once the assembly is completed, the virus releases an enzyme that disintegrated the cell wall from within, thus destructing the host cell. Or at last, the destruction of the host cell is happening. The viruses are individually and free. The more and more the virus is produced, and it is breakage of the host cell happens and it moves outwards. Next, second cycle is the lysogenic cycle. Here also we can see a cyclic procedure. First one is the phage attaches to the cell surface bacteria, and the bacteriophage is attached to the cell surface of a bacterium, and the second step is the phage DNA enters the bacterial cell. The DNA of the phage enters to the bacterial cell. Then the phage DNA integrates into the bacterial DNA. Okay, then the fourth one is the integrated propagated. And the lysogenic cycle. The lysogenic cycle allows a phage to reproduce without killing its host. In the previous cycle, we have learned that after the entering of the virus into the host cell, it tries to disintegrate the host cell and reproduce themselves. But in the case of lysogenic cycle, the phages reproduced without any killing or disintegration of the host. Here no host cell is dead. Some phages can also use the Lytic cycle, but the phage lambda can switch between the two cycles. The first step of the lysogenic cycle is the penetration. Once the virus genetic material is inserted, it attaches itself to that of the host. Some virus may not immediately start multiplying or replicating after infecting the cell. In case it does not start replicating, the virus is said to be dormant. The virus genetic material is known as the prophage, while it is in the dormant stage. The second one is the replication of the genetic material. Therefore, the cell will fall back into its regular metabolic activities and eventually prepare for cell division. The genetic material or nucleic acid replicates and the nucleus divides into two parts, each part having the same genetic information. The third step is the cell division. The cell organelles will be replicated, followed by the division of the cell body, resulting in two daughter cells, each having virus's genetic information incorporated to their genetic material. The cells have uttered normally until the virus is triggered. This daughter cell carrying the viral genetic material are known as the lysogenic cycle. Okay, in this lysogenic cycle, the cell division, that means the cell organs will be replicated, followed by the division of the cell body. When the virus or is attached to the host cell, it starts dividing themselves, the replicated themselves, and it is followed by the division of the cell body. So these are the two cycles that we had learned. So thank you.